Today we're going to talk about the electron transport chain. So we're at the end of aerobic cellular respiration. So first an overview. We talked about glycolysis and how one glucose gets broken down into pyruvate and produces some ATP and NADH, 2 net of each. And then those pyruvate go through oxidative phosphorylation in the mitochondria. And again, carbon dioxide is released and NADH is produced as well. And then the acetyl-CoA goes through the Krebs cycle or the citric acid cycle. And this is producing ATP, NADH, and FADH2. From all of those cases, the electron carriers, NADH and FADH2, end up going to the electron transport chain. And this is where we're getting the bulk of ATP produced. With the electron transport chain, the energy released from the electrons being transferred from a carrier to a carrier, and we're going to see what that looks like. The energy released allows for the ultimate production of ATP. Look at what this looks like. Okay, so here we have the cytosol, so this is where glycolysis occurs. And then we have the outer membrane of the mitochondria, the inner membrane, and we have our inner membrane space and the matrix. So complex 1 is the first protein complex, and it is called NADH dehydrogenase. This converts NADH to NAD+. So we're going to have our first technical reaction occurring here. We have ubiquinone, UQ, and this is a hydrophobic molecule, so we're only going to see it within the membrane. And it shuttles electrons from complex 1 and from complex 2 to complex 3. So complex 2 is succinate dehydrogenase. It's an actual single protein. It is only a single protein, and it's found attached to the inner membrane, the matrix side, as shown here. Complex 3 is called the cytochrome complex, and it is going to take electrons from UQ and then pass them to site C. Site C is in the inner membrane space side of the mitochondria, and this transfers electrons from complex 3 to complex 4, known as cytochrome oxidase. And lastly, we have ATP synthase. And ATP synthase, you can imagine, is what is going to be involved in the actual production of ATP. So let's look at how this is done. There we go. Okay, so same picture. And now we have my animation. So we have NADH coming in from various sections, so glycolysis, pyruvate oxidation, and the citric acid cycle. And we have FADH2 coming in. Down here we have an oxygen molecule, and they say half oxygen, and you're going to see exactly why it's half an oxygen. So you can think of it as a single oxygen. But typically we never see oxygen just by itself, and so this occurs so quickly that we call it a half O2 instead of just an oxygen. So what's interesting about complex 1, UQ, complex 3, site C, and complex 4 is they're arranged based on their electronegativities. And of course, oxygen is part of that as well. Oxygen is the most electronegative. Complex 4 is a little less electronegative, a little less, a little less, a little less, a little less, and the least electronegative. So if we think about electronegativity, we're thinking about a pull on electrons. So they're bullies if they're very electronegative. So what ends up happening is oxygen way down here is the most electronegative, and it wants 2 electrons to be happy. And so that is what happens. It steals not just one, but 2 electrons from complex 4. Complex 4 needs 2 more electrons, and it can't take them from oxygen. So it's going to turn around and take them from site C, because complex 4 is more electronegative than site C. Site C wants 2 electrons. So site C says, hey, complex 3, give me yours, because site C is more electronegative than complex 3. Similarly, complex 3 takes them from UQ, and UQ, again, is shuttling within that membrane. And complex 3 takes 2 electrons from UQ. UQ says, I want 2 electrons, so it goes back and takes them from complex 1. And almost lastly, complex 1 takes them from NADH. And we know NADH is carrying 2 electrons. Now, NADH has been oxidized to NAD+. Similarly, complex 2 can do the same thing to FADH2. So it can take 2 electrons, and these would be taken by UQ. So UQ can take them from either complex 1 or complex 2. And again, FADH2 gets oxidized to FAD. Okay, now as the electrons are moving through this transport chain, energy is being released as the electrons are being moved to different complexes. And that energy is being used to pump hydrogen out of the matrix and into the intermembrane space. And we see this in complex 1. UQ does it, not so much as a pump, but it will take a hydrogen ion for every electron it takes. And complex 4 also pumps hydrogen ions into that intermembrane space. So now what's produced is we have a whole bunch of hydrogen ions in that intermembrane space and not very many hydrogen ions in the matrix. So this is creating not only a concentration gradient, but we are seeing that battery that we talked about, where we have a whole bunch of positive charges on one side and it's negatively charged on the other side. So those hydrogen ions want to get over to the back on the inside very badly, because those positive charges are repelling, so they want to get away from each other, and they want to go to the side of the membrane where there are less of them. So they want to follow their concentration gradient. So these hydrogen ions have been pumped into the intermembrane space. And before we move into what happens next, I want to talk about how we have these electrons being pulled to a single oxygen atom. And again, we call this half an oxygen molecule. So we have the 2 electrons with 2 hydrogen ions from the matrix. So again, this increases the difference between the intermembrane space and the matrix as well. And we get the production of a water molecule. So it's important to remember that we do have water as a waste molecule. So the hydrogen ions are going to flow along their concentration gradient through ATP synthase. And end up back on the matrix side. As though hydrogen ions pass through the ATP synthase, we see the production of ATP. So how is this done? The hydrogen ions, 3 of them, bind to the basal subunit, and this is later in the note. For 3 hydrogen ions that bind, This causes a spinning of this headpiece here, which leads to the conversion of ADP plus inorganic phosphate to create our ATP, that is energy. Okay, so the process, this is where we can fill in a note. So complexes 1, 3, and 4 are organized by increasing electronegativity along the chain. This creates a pull of electrons from upstream molecules and ultimately donates electrons to the most electronegative downstream molecules. Oxygen pulls 2 electrons from complex 4. Oxygen also reacts with 2 hydrogen ions from the matrix to form a single molecule of water. For every O2 gas molecule that we breathe in, there's 4 electrons being pulled through. So you can think my animation just showed 2 electrons, and that was for half an O2. So for a full O2, we're getting that process done twice. So it's not just 2 electrons, but that's going to happen twice. The end result of aerobic respiration is the production of ATP. And we see that happening in this ATP synthase complex here. The electron transport chain does not produce ATP, but it does generate free energy released from NADH or FADH2, passing electrons to O2. This creates a proton gradient. Again, we have a difference in the H+ concentration between the 2 membranes. And this is a form of potential energy because the hydrogen ions want to get to the other side. How? The electron flow from the electron transfer chain drives proton pumps in the inner membrane, particularly complex 1 and 4. Also, as UQ accepts electrons from complex 1 and from complex 2, it's also picking up protons from the matrix. As it migrates through the membrane, it drops off the hydrogen ions and donates an electron. So recall a proton motive force where one side of the inner mitochondrial membrane has more protons than the other side. This can be used as a source of energy. So we have 2 factors at play. First, we have the chemical gradient across the membrane due to the concentration difference. So the hydrogen ions want to follow along its concentration gradient to go to the other side. The other is the protons are charged. So the positive charges are going to repel each other, and again, they're going to be attracted to the other side, which will be more negative because there's less hydrogen ions on that side. So the process is called chemiosmosis. ATP is synthesized using the energy of that gradient. This is used, as we see, in the mitochondria. It's also used in chloroplasts, which we're going to see. And it's used to pump substances across membranes. Interestingly, it's used to drive the rotation of flagella in prokaryotes. So looking at the ATP synthase itself, so this is at the very end, it spans the inner membrane. So this would be the intermembrane space. This is the outer membrane. This is the matrix. The basal unit is embedded in the inner membrane. It's connected to the headpiece, which extends into the matrix. This forms a channel for the hydrogen ions to pass through. The proton motive force moves protons in the intermembrane space through the channel in the enzyme's basal unit, down their concentration gradient into the matrix. The flow of protons powers ATP synthesis by the headpiece. So binding of 3 hydrogen ions to the binding sites in the headpiece. So we're going to have 3 hydrogen ions binding. This catalyzed formation of 1 ATP molecule, and that's from ADP and inorganic phosphate. This is the reverse of active transport that we talked about in Unit 1, where ATP was used to transport molecules against their concentration gradient. In this case, hydrogen ions are moving along their concentration gradient and making ATP. All right, lastly, your textbook talks about uncoupling proteins, and these are really interesting. So when electron transport chains exist, but there is no ATP synthase used. So you're creating this concentration gradient between hydrogen ions, between the two different sides of the membrane, but they're not flowing through the ATP synthase. Instead, there's uncoupling proteins. They provide an alternate route for the hydrogen ions to pass into the matrix. This allows a lot of heat to be released instead of actually just producing something. So brown adipose tissue, so this is fat tissue, has a lot of uncoupling proteins. Hibernating animals, like this bear here, birds, and human infants have this, and this allows them to maintain their body temperature. So that's the electron transport chain overview. We're going to have some activities where we really dive into practicing and making sure we have an understanding of this concept. Just remember that we have a coupling of oxidation of different complexes where we have a stronger and stronger pull of electrons. And that's being coupled to the oxidative phosphorylation of ATP. So we create this hydrogen pump where the energy released from electrons being passed to carrier, the energy released is used to pump hydrogen ions into the intramembrane space, and that concentration difference is used to create ATP.